So, as we've already heard, really, there are multiple approaches to antibody validation, and I take the view that antibody validation includes any sort of experiment or control which will help establish really whether the antibody is behaving as you would expect it. So, starting from very simple things like, does the antibody give the right size on a Western blot? Controls like using the no primary control we heard about. We do a lot of immunofluorescence, and I find actually it's quite a useful control. In most cases, it may well be completely blank, but the no primary is useful just to catch sort of background problems which are nothing to do with the primary antibody, and especially if it's a person who's new to immunofluorescence, or it's a new tissue cell type, or even the new part of the cell which you haven't really looked at before. It can just help you give a bit of confidence. And then, of course, you have assays like doing knockdown, knockout tissue, which we just heard about, where you can really start to tell whether the antibody or the band or whether the antibody is giving you a specific signal or not. So, and this is an example just from some of our work where we were looking at a protein called RASSF7, and we'd purchased an antibody and we didn't know whether it was going to work or not. There was only one single paper on this protein at the time, which was purely sequencing it, so we had very little to go on. So we did a knockdown, and you can see that the major band goes away in the knockdown, or is greatly reduced. But there are clearly bands up here which don't seem to change very much and are likely to be non-specific. So it gave us a very good handle on what bands on the Western were specific or non-specific. And then you can go right up to sort of high throughput approaches that I think we'll hear about in a little while. So I think it's not as simple. There is one assay which is going to validate your antibody, one approach that's going to validate your antibody or not. And of course, it's also the case that it's not, you need to validate it for each application that you're using. So antibodies may well work in one application and not another. And just an example I picked out, these are a couple of anti-GFP monoclonal antibodies that were raised by the Developmental Studies Hybridoma Bank. And in this experiment, these are cells which were transfected with GFP, and you can see the fluorescence from the GFP protein itself in green, and staining with these monoclonals here in the orange color, and you can see the overlap with where the antibody is staining the GFP protein compared to the GFP fluorescence. If you leave out the control, then you can see the GFP signal, but not leave out the primary antibody, then you can see the GFP signal, but not the signal from the antibody. So these antibodies are reported to work in immunofluorescence and also in flow cytometry and chromatin immunoprecipitation, but they don't work in Western blotting or in dot blotting. And this is consistent with the way these antibodies were raised. They were raised with whole protein, and they were specifically raised to try to generate antibodies which would recognize the native folded version of GFP so that they would be suitable for applications like chromatin immunoprecipitation. So you need to validate your antibodies for each application that you want to use them in. And if you work in a non-human species, then you also need to validate it for the species that you're using. So most commercial antibodies will be raised against human proteins, and the antibody may work really well against that human protein. But if you're, like many developmental biologists, working in mice or in amphibians, Drosophila or Arabidopsis, then you're going to have gradually more and more sequence divergence, and there's more and more chance that the antibody, or it's harder to predict whether the antibody is likely to cross-react. So you need to validate your antibody not only for the application, but also for the species of tissue that you're using. And what this means really, I think, is that it is an ongoing process. It's not something that's ever going to be finished. It's going to be an ongoing process. And or if you're at the bench, then I think many people see it as a never-ending struggle. And also, I think we've probably already heard, and I don't think it's controversial to say that some suppliers do more validation than others. Some suppliers will take it more seriously. But even the very best supplier that does the most rigorous validation, which can follow the guide we've heard about, There's no way that you can expect a supplier to test the antibody for every single application that you might use and every set of conditions in every single species that a researcher might ever want to use the antibody for. And of course, you also have every single tissue for each species. So I think when choosing an antibody, that's where it's useful to start thinking about the independent validation, which can help sort of crowdsource information which the suppliers can't generate themselves. And there are several types of independent validation that people can use to help guide their choice of antibodies. So there's contract research organizations or labs, academics labs, which will do it on a commercial basis, which will independently validate an antibody from a supplier and give it like a stamp of authority. There's antibody reviews that we'll hear about from Simon. But at CiteAb, what we really do is focus on peer-reviewed publications. And of course, as mentioned, publications are not perfect. But what we feel that they do is give a very good, if you're starting off, you want to choose an antibody to start working with and to validate in your own lab, then they provide really the best way of making that initial selection. So what we did was we realized that if it would be handy to have a resource which showed the citations that are available to antibodies to help scientists make this choice between which antibody to buy and start using and validating in their lab. So this is where we set up CiteAb a few years back. And it's really guided by two kind of key principles, I guess, which is that all the searches are done by CiteAb are done by the number of citations as a way of ranking the antibodies, that we're independent from the antibody suppliers, and that we offer a free-to-list model. And this was important because it meant we could try to capture as much of the market as possible. The goal for me coming as an academic was to try to make it as useful as possible to researchers. So if you can try to capture the whole market, then you can try to make it as useful as possible. And what that means is that because it's free to list, we've managed to generate, to capture an awful lot of antibodies. There's 2 million antibodies on, or just over 2 million antibodies on CiteAb from 100 and odd suppliers. And we have about 500,000 citations which cite the antibodies contained within CiteAb. So it's effectively a very simple search engine. You just type in the name of the protein you might be interested in. You then get a search results page, which will have some key information about the antibody. So you can see from the way I think about antibodies, the application that it's used in, the species that it's used in. And again, just ranked in this very simple way that it's ranked by citations. What's key is that we're not trying to say that scientists should just take simply 141 as being better than 140. It's important that scientists do look at use this as a guide to select a few antibodies to look at in more detail. And we provide filters so you can do this. If you've got a favorite supplier and you really want to start with them, then you can filter by supplier. Probably more commonly is people want a particular host species, they can filter by host species, or that if they want an antibody which is supposed to have been validated for a particular application, then you can filter by application. Then you just click on the antibody, and then you can go to the antibody page, which gives you a bit more information and a very prominent link to the supplier's website. We see CiteAb as a sort of stopping off point so that people can find some antibodies that may potentially be of interest and then migrate to the supplier's website to get more information than we can display on CiteAb. We also, again, it's this idea that CiteAb is a sort of resource of information. We're not trying to tell you that this one is better than that one. We're giving you information to make a decision. So this didn't all fit on one page, but we list all of the publications that we know about that cite the antibody and provide links to PubMed so you can go and have a look at them, or to the journal. And also, again, just trying to provide as much information as possible really to users, is that we have each publication has a publication page so you can see what other antibodies were used in that publication, and again link either to PubMed or to the journal to go in and look at the data and really look at it critically and see whether that antibody, the data does look like it's going to be a reliable antibody that's going to work for your application. And it is crucial, as because somebody at the bench can easily waste several weeks, and it's this time that you can never replace by choosing the wrong antibody. So I think, I try and encourage people to just spend a bit of time choosing. Use CiteAb, use other search engines, get a shortlist and make as best decision as you can, rather than order one in a rush and then spend literally weeks that you won't get back. Regretting that rush, I guess. So that's really how CiteAb works. I told you that it's free to list, it's free for scientists to use, so, and it's a spin-out company from the University of Bath, so we do need to make money. So the question then is, how do we make money? It's not grant funded. What we do is we've got a few income streams that we offer, which is crucial because it allows us to keep this independence of allowing anybody to list and of ranking by citations. So we offer companies the chance, if on their own antibody pages, to have adverts and logos if they want to promote a particular message. Once the user's made some decisions of the ones they're interested in, they can't influence that decision, but then they can start giving a constant sales message, or this example, Diagenode launched a new antibody they wanted to flag up to people. We were asked about custom antibody listing, so we do offer custom antibody listing. This is very much on a traditional pay-to-list model. We're obviously getting very good at finding citations, so we've started providing citations back to antibody suppliers. We can often know more about the publications that are using their antibodies than the suppliers, so we can provide those on a commercial basis. And also, what we've realized is we've got this huge sort of market view, if you like, or view of the whole market, and the citations which are using antibodies, and we can use that to put together data packs for antibody companies. So an antibody company may know how many sales they're making in the USA, for example, but they may not know how all the other companies are doing. And what we can do is show things like the share of citations. We use citations as a proxy for sales, the share of citations for that company in the US, and so provide information which can help them see how they're doing in individual countries. We can then generate other data from the citations, such as research area or the institutions. So if you want to launch an antibody to neuroscience, we can say, well, okay, these are the top institutes in the UK that use neuroscience antibodies most frequently. So these are probably the places you want to make sure that you're hitting, and some of them will be obvious, but some of them may not be. Also, individual antibodies, so rabbit monoclonals is obviously a lot of excitement about, but are they really being used by scientists and generating data which is peer-reviewed? We can show the proportion of citations which are from rabbit monoclonals and how that's changing over time. So the answer is that yes, they seem to be being used more and more. Sorry, that they are being cited more and more. And finally, again, from a product development point of view, if you're interested in knowing the number of antibodies that are available to particular human proteins, then we can break down antibodies in CiteAb by the Swiss Prot ID number and tell you which ones don't have antibodies that we've managed to find. Hopefully help companies focus their efforts. If there's going to be this move towards slightly fewer but higher quality, then hopefully this can help. So that's really how CiteAb works in terms of finding antibodies, and how we keep going to fund the service and keep it independent. It does seem to be useful, which is reassuring. We are getting lots of users, and most satisfying for me is that we're getting users coming back and reusing it, so that's good. The power of the internet means, of course, it's spread across the globe and is most frequently used by the countries you might predict. So the USA is our biggest market, biggest user base by a long way. But also China, Japan, the UK, not surprisingly given that we're based in the UK. And a high proportion of users do link to antibody companies and also to journals as well, which suggests that it's being used in the way that we want it to be. We're getting users from the top universities in my slightly biased view of the world. So Bath comes out on top, but also, of course, pharmaceutical companies use us, and research institutes carry out searches on CiteAb. So again, that's all nice to see it. We are getting the sort of networks that we would hope to be getting using CiteAb. The final thing I want to say is really, again, many of us are singing from the same hymn sheet, essentially, is that once you've chosen your antibody, then you still do need to validate it in your own lab. Now, my sort of personal opinion on this is that the level of validation will depend on how you're using that antibody. If the antibody is one of a panel of many markers, you probably need to do less validation than if the entire basis of the study is around a single protein, for example, as a biomarker. Then you need the levels of validation required will be different. And just the final thing as well is that scientists are very quick to moan about antibody companies, but very poor at then citing the products that they use. So if researchers cite the antibodies properly and include the code and the name, then it sort of becomes a virtuous circle. So we can then add the data to CiteAb and help other people find antibodies that are of interest. So that's our kind of closing message, if you like, or request to people using antibodies. So finally, I just wanted to really just thank all the people at CiteAb that are doing the work. Matt deserves a lot of the credit. He's actually at the back if you want to speak to him in the lights at the moment. And also the University of Bath, who funded the sort of prototype CiteAb and supported its spin out and formation as an independent company.